In this video, we'll discuss about enzyme inhibition, type of inhibitors, reversible inhibitors. In this one, different type of reversible inhibitors, that is competitive or substrate analog inhibitors, enzyme kinetic of competitive inhibitors, and drugs act as competitive inhibitors. So, introduction part. What is enzyme inhibition? Some inhibitors which interfere with the enzyme substrate complex. Some inhibitors which inhibit enzyme substrate complex or try to combine with the enzyme and stop the product formation. So, any substances that can diminish the velocity of an enzyme reaction is called inhibitor. That means when substance go and bind to the enzyme, so the velocity of reaction will be forwarded and it will be increased. So the velocity of the reaction will be forwarded and it will be increased until the product will be forming. But in case of inhibitor, what they do, in place of substrate, they will go and bind the enzyme and they decrease the velocity of that enzyme, so that's why they inhibit the reaction. There are two type of inhibitor: reversible inhibitor and irreversible inhibitor. What is the effect it causes? Reversible inhibitors. The second one is irreversible inhibitors. That means though you can inhibit the reaction, you can't reverse it. We can inhibit this reaction, we can reverse it. If you remove the inhibitor, the enzyme reaction will be normal. When we will inhibit that inhibitor, that time enzyme reaction will be normal. But in case of irreversible inhibitor, of this inhibitor goes and bind to that enzyme, the reaction cannot be processed. In this one, it will go and bind with this inhibitor, go and bind with the enzyme. This process cannot be processed further. If though you can remove that inhibitor from the enzyme, there will be deactivation of enzyme, there will be no occurrence of the reaction further. So that mechanism like irreversible inhibition. So as say, an inhibitor play a major role in enzyme inhibition. So there are the different types are there based on the type of inhibitor: reversible inhibition and irreversible inhibition. So majorly enzyme inhibition are two types: irreversible or irreversible. Reversible, again of three types, three subcategories: competitive or substrate analog. That means here that inhibition is similar to the shape of the substrate. So enzyme does not know the difference between inhibitor and substrate. Does not know the difference between inhibitor and substrate. So by mistake it will go and bind with inhibitor and form an enzyme inhibitor complex. So that's why it is a competitive. It is a competitive work. There is competition between substrate and inhibitor to go and bind with the enzyme. Second one is non-competitive, because here the inhibitor is not at all in substrate analog to, it is not in similar safe to the substrate, but somehow it will go and bind. There is no competition between substrate and inhibitor, but still it is having a major effect inhibition the reaction. Third one is uncompetitive. Other than active side of that enzyme inhibitor, other than active site of that enzyme inhibitor will go and bind and deactivate enzyme substrate complex. Enzyme substrate complex. Here deactivation. Next, irreversible. Once inhibitor goes and bind that enzyme will be totally gone. So, example like suicide inhibition group. Suicide or mechanism based inactivation, then reactive reactive substrate analog or affinity inhibitors, affinity based inhibition. We will discuss one after other. First one is reversible inhibition. So reversible inhibition is like which bind to enzyme though non-covalent bonds like hydrogen bond, hydrophobic interaction, van der Waals force, electrostatic interaction, and activity of enzyme is restored fully when inhibitor is removed. So here non-covalent bond like hydrogen hydrophobic and van der Waals force interaction and activity of enzyme is restored fully when inhibitor is removed. So that is the advantage of that inhibition. So though there is a inhibitor goes and bind to the enzyme, there is a stoppage of the reaction. Once if you remove that inhibitor, substrate will go on bind and reaction further proceed, and there are different type of reversible inhibition like competitive or substrate analog inhibitor, non-competitive inhibitor, and uncompetitive inhibitors. What is the difference between competitive or substrate analog inhibitor, non-competitive inhibitor, and uncompetitive inhibitor? With the name itself, you can make out competitive inhibition or substrate analog inhibition. Here inhibitor is similar shape with the substrate. A competitive inhibitor is structural analog of that substrate, and a chemical structure of that inhibitor resembles that of substrate and bind enzyme to at active site. So we are all aware where the substrate will go and bind to that enzyme to go and bind to that active side of that enzyme. So here there is a huge competition between inhibitor and substrate to go and bind with active site of that enzyme. So in competitive or substrate analog inhibition, the inhibitor will go and bind to that free enzyme. See here enzyme bind to substrate or inhibitor, so it will form enzyme substrate complex and enzyme and product. So here this also have a competition. Here that inhibitor will bind, the inhibitor go and bind will form enzyme. Here it will form enzyme and inhibitor complex. Here no product. Here enzyme bind with substrate, enzyme substrate complex, it producing product, but here no product, inhibited. So this way it will work. See here, competitive. So inhibitor also similar, inhibitor also similar, safe. With though you see this diagram, substrate and inhibitor both are in relation with that safe, both are relation with the safe. So enzyme does not know different between substrate and inhibitor. So whoever comes first, it will take it and form a complex. If substrate coming first, it will form enzyme substrate complex and it is producing product. This one is the product. But here that inhibitor go and bind with this enzyme, so it form enzyme inhibitor complex. There is no further reaction. The process that is inhibited, there is no product. The inhibition would be overcome by increasing substrate concentration. That means huge competition between enzyme and substrate and inhibitor towards the enzyme. So if you want to reverse this inhibition, you can to increase substrate concentration. So you if you increase the substrate concentration, so more substrate are available. So enzyme prefer to bind with the substrate rather than inhibitor. Succinate and fumarate. Succinate and fumarate. So melanate, the enzyme is succinate. This one is the melanate. Succinate dehydrogenase enzyme. So the enzyme succinate dehydrogenase. What happens? Succinate and malonate. Both are structurally similar. Only the different is CH2. This CH2 group only different. One CH2 group. So succinate dehydration is does not known the difference between succinate and malonate. So when that melanin is available, that melanin will go and bind to that succinate dehydrogenase. There is no formation of fumarate. So this is the type. So once you remove that malonate again, this succinate dehydrogenase go and bind with the succinate. If you remove, you will increase that succinate concentration. Melanate will be removed from that succinate dehydrogenase and succinate will be converted into fumarate. So coming to that enzyme kinetic, that's what is importance of discussing enzyme kinetics. So the different enzyme reaction are available with the help of enzyme kinetics. Different enzyme activities and their behavior you can access. You can see here this green color and red color. Green and red color mentioned here. So without inhibitor in the presence of substrate, how the reaction is taking place. This is the normal 1 km, normal 1 km, and this is the Vmax. Okay, Vmax half, Vmax 1 Vmax. But in case of competitive inhibitor, that km value will be increased. In the case of reversible inhibition, km value increased and Vmax altered it neutral. Vmax is unaltered. Drugs, so a treatment of like various disorder. So the drugs are mainly in reaction with the competitive inhibition. The drugs act as competitive inhibitors. So for example, sulfonamide, this is the analog of para-aminobenzoic acid and inhibition of folic acid. Uh, so we are all aware folic acid is required for making genetic material. DNA. For that, folic acid is essential. So, the part of folic acid is para-aminobenzoic acid. Para-aminobenzoic acid. So, in that concentration, the sulfonamide is similar to para-aminobenzoic acid. It will go on fit to the folic acid, and sulfonamide is used folic acid will not be useful in making of DNA. So that way that microorganism will not survive, it will not resynthesis the cell. And isoniazide, it is again anti-tuberculosis drugs, inhibit that biosynthesis of NAD and restrict the growth of that organism that causes tuberculosis. So isoniazide, not only the microorganism NAD, but in human also there in a synthesis of NAD from tryptophan. So it will inhibit the production also. So there is a huge competition between isoniazide and other substance towards that enzyme which is synthesis. Next, tugumarol. Tugumarol, anticoagulant drugs. It is structurally similar to vitamin K inhibitor, vitamin K activity, and in this formation of prothrombin for physocytic mind, it inhibit acetylcholinesterase used to treat acetylcholinesterase are used to treat glaucoma and myasthenia gravis. So these are all the drugs which are acting as a competitive inhibitors to treat these disorders, and the drugs access continuous and implement region like ibuprofen, ibuprofen and statin drugs. Cholesterol lowering drugs are cholesterol lowering drugs inhibit of enzyme that involved in prostaglandin cholesterol synthesis respectively.